Welcome to this explainer. Today, we're jumping right into a deep dive on a seriously stealthy, microscopic pathogen. It's one that has totally shaped agricultural history and, honestly, continues to pose a massive threat worldwide. We're talking about brucellosis. It's a disease caused by a fascinatingly complex bacteria, and to really understand it, we're going to have to look closely at biology, agriculture, and, yes, even global trade.
You know, it's pretty easy to think of veterinary diseases as something confined to the barnyard, right? Out of sight, out of mind. But what does a microscopic bacteria in farm animals actually have to do with the global economy? Or, for that matter, the safety of the cheese sitting right now in your fridge? Well, as you'll see, the line between animal health, international commerce, and human safety is incredibly thin. And this particular pathogen, it knows exactly how to cross it. So, here's our roadmap for today. First, we'll meet the Brucella bacteria. Second, we'll look at the agricultural threat. Third, the human crossover. Fourth, diagnosing the disease. And fifth, why global control matters. Let's kick things off with Section 1. Meet the Brucella bacteria. The bacteria up close. Let's meet our microscopic hijacker. Taxonomically speaking, DNA studies actually show this genus contains only one true species, Brucella Melitensis. But for practical, everyday reasons, we still use its traditional species names based on the host they prefer. These small gram-negative coccobacilli have a really neat trick. Under a modified Zeal-Nielsen or MZN stain, they absolutely refuse to decolor. They just pop as these bright red clusters right against the blue cellular debris. They love oxygen, they thrive on carbon dioxide, but their true defining trait? They are intracellular pathogens that zero right in on reproductive organs. Now, check out how these bacteria actually survive. When we isolate species like Brucella abortus or Melitensis in the lab, they form what we call smooth colonies. These smooth forms possess a major outer membrane lipopolysaccharide. Think of this like biological body armor. It makes them highly virulent and lets them persist and multiply right inside the host's own cells. On the flip side, we have rough colonies, like those of Brucella ovis or canis. They lack this lipopolysaccharide armor. Because of that, they're generally way less virulent and get cleared out more easily by the host's immune system. Make no mistake, though, those smooth ones are the true danger. So how does this microscopic danger actually take hold? Honestly, it's like a biological Trojan horse. First, the virulent Brucellae enter the body, often right through the mucous membranes, and get naturally engulfed by the host's phagocytic immune cells. But instead of being destroyed, they hijack the transport system. They literally ride within these immune cells all the way to the regional lymph nodes. And here's their brilliant survival trick. They survive inside the macrophages by inhibiting the phagosome-lysosome function. Basically, they completely stop the host cell's digestive enzymes from breaking them down. This leads to intermittent bacteremia, where the bacteria periodically spill out into the bloodstream, spreading and setting up camp in very specific organs. Now that we know how it works on a cellular level, let's zoom out to the farm. Section 2. A threat to agriculture. Livestock at risk. If you look here, you'll notice a pretty grim pattern. Whether it's Abortus in cattle, Melitensis in sheep, or Canis in dogs, these bacteria act as indefinite reservoirs for disease. Across the board, they cause absolutely devastating reproductive ruin. We're talking abortions, orchitis, severe infertility, and sterility. Each Brucella species tends to target a specific animal host, acting as a highly specialized agent of agricultural destruction. In endemic areas, this completely decimates herd fertility and drastically tanks milk production. But why? Why does this bacteria go after the reproductive organs so specifically? Well, the answer comes down to a specific chemical called erythritol. This is a polyhydric alcohol that is naturally found in incredibly high concentrations in the placentas of cattle, sheep, goats, and pigs. For Brucella, erythritol is literally a superpowered growth factor. The bacteria are chemically drawn to the placenta, the mammary glands, and the epididymis simply because it provides the absolute perfect, nutrient-rich environment for them to explode in numbers. And the objective stakes for a farmer, they become painfully clear when we look at this number: 5. In cattle, an infection with Brucella abortus often leads to catastrophic abortion storms in susceptible herds. And this tragic event almost always strikes right after the 5th month of gestation. The sheer immense concentration of bacteria excreted in fetal fluids at this time, which can often last 2 to 4 weeks, massively contaminates the environment. It pretty much guarantees the rapid spread of the disease to the rest of the herd. But the story doesn't end out in the fields. Let's move to Section 3. The human crossover, zoonotic transmission. We've seen how perfectly specialized Brucella is at hijacking animal biology, but what actually happens if this animal pathogen crosses over to us? Humans are highly susceptible to B. abortus, B. suis, B. melitensis, and even occasionally B. canis. And when it makes that jump, it brings an entirely new set of severe systemic problems.
In humans, the disease is notoriously debilitating. Historically, it's been known as undulant fever or Malta fever. Now, unlike in animals, abortion is not a typical feature in human infections. Instead, patients suffer from fluctuating pyrexia, basically a fever that rises and falls in rolling waves, along with profound malaise, chronic fatigue, and really intense muscle and joint pain. Osteomyelitis, an infection of the bone, is actually the most common complication. And the species matters big time here. Infections caused by Brucella melitensis from sheep and goats result in particularly severe systemic disease.
So, the crucial point for us is understanding how we actually catch it. Because luckily, human-to-human transmission is incredibly rare. We get it through direct contact with infected animal secretions. The everyday risks are shockingly common. Drinking raw milk or eating dairy products made from unpasteurized milk are major sources of infection. It can also enter right through simple skin abrasions for those handling animals, or even be inhaled. And in a fascinating, slightly ironic twist, because it's so easily aerosolized and so highly virulent, the scientists studying it are actually at extremely high risk. Brucella is a notorious cause of laboratory-acquired infections. Because it is such a stealthy and dangerous pathogen, identifying it is an absolute top priority. Let's look at Section 4, diagnosing the disease, identifying the threat. You literally cannot just swab an animal and look at it on an open lab bench. Because of the extreme risk of laboratory infection we just mentioned, diagnosis requires incredibly strict safety protocols. Processing any clinical specimens, like cotyledons or fetal contents, has to be done inside a specialized biohazard cabinet. Scientists have to culture the bacteria on highly nutritious media like Columbia agar supplemented with 5% serum. Then they have to incubate it at 37 degrees Celsius in a 5 to 10% carbon dioxide atmosphere for up to 5 days, just to confirm it's actually there. But as you can guess, culturing the bacteria is way too slow for real-world farming. To manage herds, veterinarians rely on a layered toolkit of serological tests. We have the continuous monitoring of bulk milk using the Brucella milk ring test, which lets us keep an eye on entire dairy herds at months. For rapid individual screening, the Rose-Bengal plate test is the standard go-to. And if an animal flags positive, we bring out the heavy artillery, rigorous confirmatory tests like the complement fixation test or modern ELISAs. These tests are the literal frontline defense in figuring out exactly which animals are hiding this invisible threat. And that frontline defense is totally necessary for a much larger worldwide strategy. Section 5. Why global control matters. Global eradication. Listen to this quote from the source material, because it really hits the nail on the head. Infected animals serve as reservoirs of infection, which often persists indefinitely. Treatment of cattle with brucellosis is simply not practical. It just doesn't work. The bacteria hides way too well inside the host's own cells, and the animals shed huge amounts of it into moist environments where it can remain viable for months. Because you literally can't cure the herd, eradication is the only viable path forward. So, what does actual eradication look like? Well, it requires massive coordinated action. Countries that have successfully reduced or eliminated bovine brucellosis rely on rigorous national test and slaughter programs. Sadly, any animal testing positive is removed. Second, strict serological testing is an absolute requirement for international trade just to prevent reintroducing the disease across borders. And finally, during the early years of an eradication scheme, strategic vaccination of young replacement animals is absolutely critical to build up herd immunity. Historically, we relied on attenuated vaccines like S19 for cattle or Rev1 for sheep and goats, and they were great for young animals, but they had a major flaw. If you vaccinated mature animals, they developed persistent antibody titers. Essentially, they triggered false positives on our tests, completely confusing our surveillance efforts. Enter the RB51 vaccine, a truly brilliant scientific advancement. It's a stable, rough mutant of the bacteria. It induces cell-mediated immunity without leaving the specific antibody footprints that ruin our global data. It's a real game changer because it allows us to protect the animals while keeping our international surveillance perfectly pristine. Which brings us to the ultimate takeaway of this explainer. We've traced this disease all the way from a little red cluster on a microscope slide to a devastating abortion storm on a farm, to severe undulating fevers in humans, and finally to the strict international testing protocols required to stop it.
Think about it. If a single microscopic bacteria can disrupt global trade, ruin agricultural economies, and directly threaten our food supply and human health, can we ever afford to stop monitoring zoonotic diseases? The biology of Brucella proves that our global health is deeply, deeply interconnected, and total vigilance is our only true defense. Thanks for joining me, and I'll catch you in the next one.